Welcome to Omni Academy. This is Maria, and today we are starting our second lecture on development of eye. In the last lecture, we studied the time, the outcomes, the sources, and the master regulator gene for eye development. In today's lecture, specifically, we will be focusing on the two main sources of eye development, which are neuroectoderm of diencephalon and surface ectoderm of head. And we will be seeing that how these two sources are contributing to develop human eye. So let's start with today's lecture, which is early morphogenesis of eye. As we studied in the last lecture that it is the 22nd day or the fourth week of gestation when this process of development of eye starts in an embryo. Now let's see that how this process begins actually. So it is around the 22nd day when the neuroectodermal diencephalon produces outpouchings called optic grooves. So this is diencephalon, this is neuroectoderm of diencephalon, and around the 22nd day this neuroectoderm of diencephalon is producing shallow grooves which are known as optic grooves. So the formation of optic grooves is the first step in the development of eye. So now let's move to the second step. As these grooves grow, they become bubble-like structures known as optic vesicles. So you can see in this diagram that now the optic grooves are growing towards the surface ectoderm. And as they are growing towards the surface ectoderm, they are becoming bubble-like in structures. So these bubble-like structures are known as optic vesicle. So the second step in the development of eye is the formation of optic vesicle. So now let's move to the third step. When the optic vesicle touches the surface ectoderm, it induces the formation of a lens placode. So you can see in this diagram that the optic vesicle is now touching the surface ectoderm, the ectoderm which is present above the optic vesicle. Because of this touching is now converted into a lens placode. So the third step in the formation of eye is the lens placode formation. So now let's move to the next step, which is both vesicle and the placode invaginate, fold inward at the 28th day. So you can see in this diagram that the placode and the vesicle both are invaginating. This invagination turns the vesicle into a double layered optic cup and the lens placode into a lens vesicle. Let me show you this in diagram. So you can see that this was the optic vesicle, but because of the invagination it has now two layers, the inner layer and the outer layer, and this has now become an optic cup which has two layers. This is the inner layer and this is the outer layer. So these are the structures that are formed from the invagination. And what these structures are: lens vesicle and the optic cup. Optic cup is formed because of contribution by the neuroectoderm. But the lens vesicle is formed because of contribution by the surface ectoderm. So this is the role of the neuroectoderm and the surface ectoderm in the formation of eye. So now let's talk about the optic stalk. So diencephalon and the optic vesicle, or you can say the optic cup, has a connection between each other. This area is the connection between the diencephalon and the optic cup. This area with the passage of time narrows down and we call this area as optic stalk. So now let's talk about the derivatives. So the optic cup in this process of development of eye will form retina, iris and ciliary body. But the lens vesicle in this process of development of eye will form lens, conjunctiva, cornea and eyelids epithelium. And the optic stalk in this process of development of eye will form optic nerve, optic chiasm and optic tract. So now let's talk about the choroid fissure. Choroid fissure is a groove which is present on the inferior aspect of the optic cup and the optic stalk. This is choroid fissure, and the significance of it is that it allows the hyaloid artery to enter in front of the optic cup and supply the lens vesicle, or to supply the developing internal structures which are present in front of the optic vesicle or the optic cup. So this is choroid fissure, and this is allowing the hyaloid artery to enter in front of the optic cup and supply this structure which is the lens vesicle. So this is the developing internal structure in this case. The choroid fissure closes by the seventh week, and this closure of choroid fissure converts the mouth of the optic cup into future pupil, from where the light enters. Intraretinal space. There was a space between the two layers of the optic cup, which we call as intraretinal space. This space also obliterates by the seventh week. So now let's talk about the hyaloid vessels. Firstly, let's just talk about the course of the hyaloid vessel. Proximally, hyaloid vessels are transported within the choroid or optic fissure in optic stalk. Distally, they are transported in the vitreous body. Proximal part of the hyaloid vessels become central artery and vein of retina in the adult, and the distal part of hyaloid vessels degenerate, leaving a hyaloid canal in the vitreous body. So this is the fate of hyaloid vessels and the course of hyaloid vessels. So this choroid fissure closes by the seventh week, and this converts the optic cup into a circular mouth, and this circular mouth in adult is known as pupil through which the light enters. This is intraretinal space which is present between the inner and outer layer of the optic cup, and this also obliterates by the seventh week, and the two layers also fuses by the seventh week. So let me just summarize the early morphogenesis of eye. So the first step in this was the formation of optic groove. Second step was the formation of optic vesicle. Third step was the formation of lens placode. Fourth step was the formation of optic cup and lens vesicle. And we have discussed that optic cup and lens vesicle were formed because of the process of invagination. And then we have discussed about the optic stalk, and after that we have discussed about the derivatives of optic cup, lens vesicle and the optic stalk, and the last step was the formation of choroid fissure. And after that we have discussed about that when this choroid fissure closes and how the pupil after that closure is developed. And we have also discussed about the closure of the intraretinal space, which in turn result in the fusion of the two layers of the optic cup. And we have also discussed about the course and the fate of the hyaloid vessel. So this was today's lecture. See you in next lecture.